All right, now let's take a look at these changes in membrane potential that we get in response to that neurotransmitter binding, right? Remember, we had neurotransmitter coming in, binding to receptors, leading to ion channels either opening or closing, and that's going to lead to some change in membrane potential, okay? What I want to point out about that membrane potential, that membrane potential change that we get is that that is a graded potential, okay? So drawing your attention to this figure right here, you can basically see that process happening, right? So we have a stimulus coming in, which is the action potential from the presynaptic cell. That has led to neurotransmitter release, which has led to this change in membrane potential in the postsynaptic cell, okay? We have a name for this change in membrane potential. It is called a postsynaptic potential, or a PSP, okay? And like I already described, but is verbalized more, I don't know, better in these words right here, a PSP is a graded potential that's going to happen in response to neurotransmitter release, all right? We have two main types of PSPs that can happen in our postsynaptic cell. The first is an excitatory postsynaptic potential, or EPSP, okay? An excitatory postsynaptic potential is a PSP that is depolarizing, okay? So when we get a depolarization of the postsynaptic cell, we call that an EPSP. Take a second now to think about how you could get a PSP. Like, that's kind of a vague question. So like what kind of ion movement would lead to an EPSP? Sorry, so take a moment to think about that. Write down your answer. Okay, so you can get an EPSP either by having positive ions flow into the cell, right? Because that's going to make the membrane potential more positive. Or you can have negative ions leaving the cell. If you think about the ions that we have around our neurons and the concentrations that they usually occur in, you might remember that the positive ions, the cations that are going to be able to come into the cell are going to be sodium and calcium. And the negative ion, the anion that we have is chloride. So it would be chloride flowing into the cell, which may or may not happen depending on what the membrane potential is. Because remember, chloride equilibrium potential is like -63. Okay, anyway, moving on. Our second type of PSP is an inhibitory postsynaptic potential, or an IPSP, okay? An IPSP is a PSP that is hyperpolarizing, okay? So now just take a moment to think about this question. How might you get an IPSP to happen in a cell? So just think about what ions would be involved, write something down. Okay, so IPSP should happen either by getting cations to leave the cell, right? If we have positive ions moving out of the cell, then the inside of the cell becomes more negative, right? Or we can get negative ions coming into the cell. And if you think about the actual ions that we have around our cells and the concentrations that they're present in, that would typically happen if we have potassium leaving the cell, right? Remember, we have a high concentration of potassium inside the cell. Or if we have chloride coming into the cell, okay? All right, last quick question. Are these EPSPs or IPSPs? So think about that for a second. Okay, good. I assume that you answered that right. So these are EPSPs, excitatory postsynaptic potentials, because they caused depolarization, okay? All right. So now let's take a look at what happens to PSPs once they are, after they're generated at the postsynaptic neuron, okay? I wanna remind you that we have a PSP, the PSP is being formed near where the neurotransmitter receptors were, because that's where the channels are as well. So the PSP is being formed there. And I want to remind you that that's a graded potential. And I want to remind you yet again that graded potentials are going to degrade with distance. Okay, and that's what's shown here in this figure. We have our graded potential being formed right here in response to the neurotransmitter that has been released from the presynaptic cell. It's going to be a certain size here, and then as we travel, as that PSP travels along the cell body, remember it's going to travel in all directions. It's going to spread out in all directions, just like throwing that pebble into the pond, right? Those ripples are going to be bigger towards the center, and then they're going to get smaller as we go out from there. Same thing is happening with this PSP. It's going to be big here. It's going to ripple out, so to speak. It's going to spread and degrade as it spreads, okay? Once it reaches the trigger zone of the axon of the postsynaptic cell, that postsynaptic cell is basically going to make a decision about whether to fire an action potential or not, right? So if the PSP reaches threshold, if it's big enough to reach threshold by the time it reaches the trigger zone, then we're going to get an action potential. If it's not big enough by the time it reaches the trigger zone, then we're not going to get an action potential. So looking at this example here, we have our PSP that has started at this size. It's degrading as we move along the cell body. By the time we get to the trigger zone, it is too small. It is below threshold, and we're not going to get an action potential, okay? If a PSP is not big enough to trigger an action potential, we call it a sub-threshold EPSP. Sorry for that flashing. In contrast, we can look at this situation where we're starting with a larger EPSP. Okay, see how it's larger than this one right here? It's larger, and it's going to degrade over distance, just like the other one did, right? So it's going to degrade over distance, but in this case, even though it has degraded, it is still big enough to reach threshold by the time it reaches the trigger zone. And in this case, we get an action potential, okay? This kind of EPSP, one that is big enough to trigger an action potential, we call a supra-threshold EPSP, okay? So sub means under, supra means above, under threshold, above threshold, all right? Okay, so that basically wraps up our discussion of the response that we get in the postsynaptic cell during synaptic transmission. Here are your learning goals. Here's your vocabulary. See you in the next video.